Acho que agora não.
Pessoal, me avisem se vocês estão me vendo.
Sim.
Okay. So, I'm Laura from Universidade Franciscana. So, welcome everyone to Nano Network, and today we are very pleased to have a lecture on carbon allotropes in plant and human health, which will be present by Professor Dr. Bernard Westermann from IPB Halle, Leibniz Institute for Plants Biochemistry. And so I will invite here now Professor Cristiano Holding from Universidade Franciscana to introduce for us Professor Bernard.
Hello everyone, good afternoon, boa tarde a todos. É prazer para mim apresentar o Professor Westermann. For me, it's a pleasure to introduce Professor Westermann, my friend Professor Westermann from Leibniz Institute of Plant Biochemistry, Halle. He will give a short talk for us today. Professor Westermann graduated took his PhD at the University of Paderborn a long time ago, and after that he moved for the postdoctoral fellow at the University of Toronto, Canada, and later he returned to Paderborn, and later on, since 2004. He is the group leader of biofunctional synthesis department of the bioorganic chemistry at the Leibniz Institute of Plant Biochemistry in Halle. Professor Westermann has a long curriculum of almost 6,000 citations, working mostly in MCRs, multi-component reactions. It's editorial board, take part in editorial board on different journals, and actually he is a partner in our project internationalization project on our institution. I was there. I was visiting him from December until February, and now he's hosting one of our PhD students, Gabriela Sangoy, which having his PhD sandwich at university. Bernie, as I said before, it's my pleasure to have you here. We'll look forward to hear from you.
Okay, dear Professor Laura, dear Professor Cristiano, dear Cristiano. So it's my pleasure to have a talk on the title you mentioned already, carbon allotropes in plant and human health. And it's my pleasure to be a partner of the internalization program at the UFN. So I'm very, very pleased about this. So I visited Santa Maria already a couple of times, and it's a very, very pleasant city. And I'm very, very humbled to be allowed to travel this beautiful country and this beautiful area in the south of Brazil. So today's talk will be some literature overview, which will be on carbon nanotube growth enhancement. So we have done some work in this context, but we are not fully equipped in this one. And I will give also a talk on multi-component reactions on fullerenes, where we already have some very, very nice results. And in this case, I'm talking about some practical things we could already accomplish in this one. But before starting, I would like you to show where the Leibniz Institute is placed and where I'm coming from. So this is the province in the middle of Germany, so a little bit south of Berlin. This is the province of Sachsen-Anhalt. So you here see the bigger picture out of it, and this is the beautiful city of Halle, where I'm coming from, and where this work, or most of the work, actually took place, and which and the Institute Cristiano visited. So the most famous son of the city is Georg Friedrich Händel, a very well-known composer. So I asked a couple of times also being in Brazil if you know Friedrich Händel, and a couple of people mentioned, no, we don't know him. But as soon as I'm mentioning to you that if you know the anthem of the European Championship, also then you know most likely the music of Handel. So Handel has written the anthem of the European Championship Cup, and this is where you might know his music. So also it's a very, very nice and lovely city. So here you see the marketplace. So it's quite an old city. Also the university belongs to one of the eldest in Europe. It's called the Martin Luther University because in the area Martin Luther used to work and to pray a lot let's say 500 years ago. Halle is also hosting the German Academy of Sciences, so a very, very beautiful building where, let's say, some outlines for science is being created, especially especially in Germany. And this is the institute where we are working. So these red roof buildings over here. So we are directly integrated into the big campus of Halle. So here you see chemistry, pharmacy, biology. So all the natural sciences are located in this area, and we are just a central part of this setup over here. So now I would like to start with some overview, and it's titled by Engineering Plants with Carbon Nanotubes, a sustainable agricultural approach. So what agriculture is facing in these days or in these times is really a wide range of challenges. So climate change is one of the hot topics, plant pathogenic organisms, infectious crop diseases, soil nutrient deficiency, crop yield reduction, lack of awareness of genetically modified crops. So these are some facts which we really have to deal with in these days, and especially in Europe and some other countries. We also have to deal with the lack of workforce, which might not be so eminent in Brazil, but in Europe and also the United States and Canada, this is also a big topic when it comes to agricultural work or doing work on the farms. And so all of these facts, or there are even more, they threaten to destabilize agriculture sustainability. So this is the source I've taken for this one, so please have a look. It's a very, very nice overview, and some of the things I'm referring to in this from this article. Sustainability, this is one of the major facts in these days. So we try to be sustainable. We need to be sustainable to save our planet, and especially in the agricultural field, this is a big fact. And we already see that the climate is changing, and that we have some changing conditions, and it will continue to develop, as mentioned over here. So, and what we have also experienced that we have some really extreme environmental conditions, and also the frequency is really becoming quite alarming. So, and with this we have salinity, so salt stress to the floods. We have drought stress. Not only drought stress, but also we have floods. And I know in particular that in the south of Brazil, you have been affected heavily by huge floods in recent times. And we also have to deal with high and low temperatures, also affect in Santa Maria, as I've experienced on my own. So temperatures beyond 40 degrees is not something I'm used to so often, and which causes not only some problems to me, but also a lot of problems to the other people. So all of this can really cause extensive annual reduction of the overall crop production. So it's about so numbers are even now going up into 30%. So 1/3 of the harvest is being lost to all of these facts, and explicitly wheat, maize, barley, soybean are heavily affected by this. So the major plants, the major carriers of nutrition are affected by this a lot. And so the quality is also decreasing, and this is really giving an alarming issue for the food security globally. And as well that we have a quite increasing population on our planet, and so we really need to take care of these things. So what has been done in the history? Yeah, so not only in previous decades, but also in history. So crop rotation was really a very delightful way to overcome certain problems with some deficiency in the supply of plants. Then we have the chemical inputs. So chemical inputs have been used, fertilizers, pesticides. Conventional plant breeding has been done a lot. And this later on developed for urban farming, precision farming. Precision farming means now where artificial intelligence comes into play. So what is about having a very nice overlooking, for instance, by drones of the fields, and then really to do by pattern recognition and all of these things to go really to precision farming, which is also one of the big facts in the future. And of course, you know that the breeding is not done by traditional things anymore, but we are also now going to targeted breeding, so which means we are also now using genetically modified organisms, and you know that the acceptance of these is not as it should be. So some people are quite reluctant to use these products when they're coming from GMOs. So what has been done by farmers to adopt to all of these things? So they have used really in large amounts agrochemicals. Yeah, and you know as well, and this is not a problem in Europe or North America. This is also a problem I would say worldwide. This has caused really severe environmental problems, air pollution, soil quality. I don't have to mention all of this. You know this from your own experience. You know this from the news, and so this is nothing which is really new. And what is also interesting that chemical fertilizers, they really have a low efficacy. So they vaporize, they leach. This is also one of the facts. And interestingly, by using fertilizers or maybe an excess of fertilizers, the production costs really raise. And this is also one thing the farmers do not want to have. So they do not want to have costs ever rising. They really want to cut this really down. All of this leads to the final decision that really the agricultural sustainability has been really diminished in recent years. Yeah, so I wouldn't say is lost, but really has been diminished or has been decreased quite substantially. So we need to have new approaches. Yeah? So I mentioned one case is AI, so artificial intelligence also may improve the efficacy. But what about new agrochemicals? And interesting now here, the nanotechnology comes into play. So the nanotechnology is really a promising candidate for sustainable agriculture. So we can really underlie the transformation of traditional to precision farming by the use of nanomaterials. And by this, you can have really a well-balanced strategy by monitoring of the environmental variables. Yeah, so by really having overlooked the huge areas and then apply some modern techniques, or we can just have the implementation of precise controlled actions in response to environmental conditions. Yeah, so by this surveillance, we can really have a very detailed insight into all of these facts, and this is what nanotechnology is good for or could be developed in the future about these ones. Yeah. So as mentioned over here, it really has the potential to improve crop yields while maintaining the ecological balance, the environmental sustainability, and economic stability. Yeah? So you might say this is also wishful thinking, but as you will see in the next slides, some of these really have been accomplished already. And for this, we are out for a very nice, let's say, addition. I wouldn't say you can really exclude all the other things in the future, but it will be a very nice addition to overcome all of these problems. Here you see these nanoparticles. So in this case, I'm referring to single-walled carbon nanotubes or multi-walled carbon nanotubes. So if you apply them to plants, what you see that for instance here they interact very nicely with crops. Yeah, so they can enter the roots, they can also enter the leaves, and you can analyze it very, very nicely. And by this you can see that, for instance, the yield of the plant is really increasing. So the size normally increases, and also some other facts are quite advantageously to plants. So you have seed germination. Yeah, so here you have the seeds, and what you can also see is, for instance, that these carbon nanotubes, they can easily enter these seeds. So the germination is more efficient, is fast, is overcoming also some problems in case of salinity or drought. So you can overcome these problems really by treating seeds with these carbon nanotubes. You can also have it you can also use it as an antibacterial agent. So, for instance, they can disrupt the cell membrane in certain cases, and by this they can really then start the production of reactive oxygen species, and by this you can then damage the cell quite drastically. You can also deliver genes. For instance, here you have these nanotubes, and you can easily attach whatever system to these nanotubes, and they can be used as a carrier. And you see over here these genes just to modify the plants. So genetically modified organisms can be produced by these nanotubes quite easily as well. You can implant sensors. Yeah, so as seen over here, and by these sensors, so also here the nanotubes are used as some carrying system, and by these sensors you can, for instance, have a very nice environmental surveillance of an entire field, of an entire region. And what is also very nice, you can also affect the defense mechanisms of the plants very nicely. Yeah, so the stress on UV irradiation is decreased. The oxidative stress is being reduced. So the release of reactive oxygen species, the plant becomes more resilient towards drought and salinity. So all of these things are also nicely can nicely be observed when you apply these nanotubes to plants. Yeah? So this is some summary of the diverse applications of carbon nanotubes in the agricultural field. Also, history-wise, it's quite interesting. So it started. So the first publications came out in 2008. So you see, the entire field is not very old. And it has been shown that, for instance, if you have carbon nanotubes, non-functionalized ones, they can really affect the root elongation of cucumbers. Yeah, or here it can also seen in a very impressive way, and I will show it later as well, that, for instance, they can penetrate the tomato seeds. Yeah, so you have the control, and here you have the one which has been treated with carbon nanotubes. So the seeds within the same time frame are getting bigger, and the germination is really enhanced, or is really improved a lot. Yeah. So and all the facts I mentioned before, you can see over here. So the toxic effects of carbon nanotubes on certain cell types has been observed. So really, you can go on, and you will have this presentation later on. So I will just share this presentation with you. So you can just have a look at all of these ones, and you can see that the increase of further knowledge how to use these carbon nanotubes is really increasing a lot. Yeah. So and you will see some more you will see some more impressive pictures within a second. Yeah. So here you still have the so here you again have the schematic view. So how the seeds and the carbon nanotubes, if they're treated within the same medium, can penetrate. You have the enhanced germination, and then the shoot and the growth of the plant is really increased a lot, and this leads to really better biomass production. Yeah? So the root length is increased. You have more biomass being produced. The biosynthetic rate goes up. The transpiration rate goes up. So the overall production of these plants is really increased a lot. Yeah. So here you see, for instance, some nice pictures. So within the same time frame, here you have the control plant. And here you have a plant which has been treated with single-walled carbon nanotubes. Interestingly, so in this study, they also use quantum dots. So you see that more or less the yield of the biomass production is about the same when compared to the controlled. But only but if you only treat it with this carbon nanotube, you can see really a drastic change within the plant. Yeah. So you can also see this one here in picture C. So the roots, the growth of the roots is also highly dependent on the carbon nanotube concentration. Yeah. So as you can see, when you have found the optimum, you really have a very, very nice increase in yield, in biomass production, root length production, and whatever. But if you go beyond this, then simply the plant dies. Yeah. So this you can see with single-walled carbon nanotubes and also with multi-walled carbon nanotubes. So on different plant species, spinach and lettuce, it can be seen. So as always, yeah. So you have a nice, or let's say a striking boundary between the positive and the negative side of using these materials. Yeah. But striking, if you have found the right condition, then the growth is really quite. Then the growth is really increased a lot. Yeah. So here we have some other examples. They also compared natural multi-walled carbon nanotubes. So when these carbon nanotubes are produced from other carbon origin, then they're called natural multi-walled carbon nanotubes, or synthetic carbon nanotubes when they, for instance, produced by vapor deposition. So what you can see that here in this case here the natural carbon nanotubes also behave the same like the amorphous carbon, and that the synthetic multi-walled carbon nanotubes is really more or less the same as with the control. Yeah. So here you also see that still that you have these effects, but still these effects need to be studied. These effects need to be determined, or the boundaries of these effects need to be determined in the future. And this is a big topic we have to deal with in the future. Yeah. So here again, you see some striking differences, as I mentioned to you. So when you have found the right condition, the biomass production is really quite drastically. Also, the watering effect is quite interesting. So if you have really, let's say, or what is also assumed with these carbon nanotubes is that the water content normally in these plants is much higher. So even under drought conditions, the content of water is much higher. So you can imagine that the plant grows much faster and much better under these additional addings of the carbon nanotubes. Yeah. So here you see again these pictures what I've shown in small in the beginning. Yeah. So this was very striking to me. Here you see the standard. So you just have a normal seed of a tomato plant, and here you just have the seed treated with carbon nanotubes. So if you, for instance, or just looking at these chambers without the addition and with the addition of carbon nanotubes, how, let's say, the plant really explodes. Yeah. Also the days of germination can be really reduced quite drastically. And again, if you have some facts within your close environment that really hamper you, for instance, from having a really good development of the plants, you can treat it with these carbon nanotubes, and then you may run out of these problems, which are joined up with these drastic conditions of drought and salinity, stress, and so on and so on. So this is really a big fact what we have to deal with in the future. So they might serve as a platform for agri-nanotech revolution, for a so-called agri-nanotech revolution. It's more sustainable than using pesticides or other chemicals in the past. But still, we have to gain a better understanding of the mechanistic pathways. So this is really still quite unclear, or I'd say it's really in an infant state. And we really need urgently and quite fast more studies to have or to be allowed to give an answer on the impact of these carbon nano particles. Yeah? So the positives are very, very striking. Yeah, so we can just substitute old pesticides and herbicides. We can come to new pesticides and herbicides. You know, the glyphosate story, that this is really a big problem in so many countries. You can have genetic, or you can really improve the genetic engineering of plants. It can also be used for the delivery of agrochemical compounds which have not been suitable before because of solubility reasons or whatever. Here you can have these engineered nanomaterials, or let's say carbon nanomaterials, to be used for this delivery. You can also sensor, which I haven't pointed out in detail, but I think the impact is quite clear. When you also deliver sensors to these plants, you can monitor so many aspects, for instance, like soil conditions, salinity, and drought, and whatever. Yeah. And you can really preserve the identity of the plant. Yeah, so this is also very interesting for the future. So the future is bright using nanoparticles, but let's see what we can achieve by further studies. Yeah, now I'm changing from this overview to, let's say, practical examples, to a practical example what we did in our own lab. So now we are changing to fullerenes, because we did this work on fullerene. You have seen that also fullerene have the same positives like these carbon nanotubes, so plant growth enhancement, seed germination, and so on and so on. But you can also use it for human health applications. So it can be used as contrast reagents, as antivirals. You can deliver drugs more easily. It can be used as an antioxidant, and what we checked was here just to use them in the photodynamic therapy. So using so being allowed to treat cancer patients. The problem with the fullerene, also with the other carbon nanotubes, is the solubility. The solubility is really striking, is really low, and this really hampers a lot the application as drugs or as carriers in the future. So here you see some of the solubility, and in particular, if you go for cells, humans, animals, or whatever, here you see the very, very low solubility within water, yeah, which is really too bad. And so you need to modify these fullerenes and to gain some solubility. And this is what we carried out in this nice paper over here. So we wanted to modify the fullerenes by multi-component reactions. So I will come up to these abbreviations. So Passerini 3-component reaction, UGI 4-component reaction, I will explain in the future, to have these modified fullerenes, which are very much improved in their water solubility and can be used also in some medical applications, like I mentioned in the photodynamic therapy. So this work was carried out also with the aid of Brazilians. So in this case, it was done at the University Federal of Santa Maria with our colleague over there. And just for the beginning, for the explanation, so multi-component reactions. So in most of the cases, what you study in chemistry are these sequential reactions. So you just have a starting material and a product. So this is a one-component reaction. You may also have two-component reactions where two of these are going to be reacted to give you one product. So this is an addition reaction, for instance. But if you, for instance, change to 6 compounds added at the same time, giving you one product, for instance, then you have a 6-component reaction. Yeah? one of the leading persons in studying multi-component reactions is Alex Dömling, and this schema just stole from him from these sources here. Give you an explanation to the UGI 4-component reaction. So in the UGI 4-component reaction, you're using aldehyde, carboxylic acid, an amine, and an isonitrile. And what comes out of this is a peptoid. Yeah. So in comparison to a peptide, one of the amide groups is a tertiary amide group. So here you have a substitution. The peptide just has NH bonds here, and this is the reason it is called a peptoid. Yeah. And the UGI reaction, when it comes to sustainability, is also a very nice reaction. It's almost an ideal reaction. So you can use it under ambient conditions. So almost any solvent, water, alcohols, everything can be used in most of the cases. You just stir it at room temperature, then it's fine. You can do it really in a combinatorial way. So you really can use a lot of these different components. And interestingly, the only formal byproduct is water. Yeah? So it's very atom economic and step economic. So one step, and with the exception of water, all the atoms are being incorporated into the product. So beautiful reaction. The Pasolini reaction, which is slightly elder than the UGI reaction, found out by this Italian chemist Pasolini. He is using 4 components, so he doesn't use the amine, so only an aldehyde, carboxylate, and the isonitrile, and what comes out is the peptide. So one is an ester bond here, and the other one is an amide bond. Yeah, so, but this is just a 3-component reaction. To have material ready for this reaction, so we took the fullerene, treated it with this very, very nice material, which has been described a couple of years, some 30 years ago, and then you have this addition here to the fullerene, and this ester is then can be obtained in very nice yield as the sole product. You just do the cleavage of the ester, and then you have the carboxylate, which then can be used for further reactions with the UGI reaction. Interestingly, you can nicely follow this reaction. So if you have the naked fullerene, for instance, dissolved in toluene, this is has this nice color. So it turns a little bit reddish when you have the ester being formed and when the ester is being cleaved. So then you have the carboxylate, then this brownish color appears within the solution. So you can also nicely follow if the reaction has been completed or not. Yeah, and now I'm going to point out the results we had here with the Pasolini 3-component reaction and with the UGI 4-component reaction after cleavage of this ester, and using the fullerene carboxylate as one of the starting materials. So we started by using normal aldehydes, isonitriles, and aliphatic amines, and we didn't obtain any product. So, but then my Brazilian PhD student, then he easily found out that when you use aniline in these cases, that you can really obtain in very, very nice yields the appropriate product. Maybe some stacking of the surface of the fullerene and of the aniline is necessary to achieve this very, very nice reaction. Here is just a table of some of the UGI products we synthesized. So you can see here this peptoid structure, so the substituted amide bond, and here the secondary amide bond. We also used some very, very nice isonitriles with a functional handle with a convertible isonitrile, so the amide bond can easily be cleaved into this pyrrole, and then you can do the substitution at this point very easily. We attached complete peptide chains in here. So we had tetrapeptide, hexapeptide. We could even go longer. And interestingly, if we use a functionalized bis-aniline, we can also bridge these fullerenes. Yeah, so we can have these dimers produced really in quite some reasonable amounts, 42%. This is just for the synthesis. Yeah. Passerini, we did as well. So with the exception or with the exclusion of amides, you can obtain these peptides quite nicely. Here, you see just a variety. So we have these PEG chains in there. So by this, we have really nice soluble products. We can also have these amines at the end, or protected amines. We can add carboxylates. So we can do also further passerine post-Ugi reactions with these compounds just by cleaving here the ester or here the amine. We can also add dyes, so to apply them later on in biological studies and just studying the fate of these molecules. Yeah, so what we did as well to play around a little bit with these molecules, we wanted to have these dicarboxylates, so bisfunctionalized fullerenes, and this is a bit problematic. So you have certain regioisomers which can be formed very easily if you just do it by this approach we did which has been reported by Wilson, as mentioned before. So then you can have a couple of these double bonds being treated in this 2+1 cycloaddition, giving you the cyclopropane. So it's not very feasible to do it just right away. So you have to do some tethered approach. So here you see one of these starting molecules. So we have two malonates, and the malonates are just tethered by this aromatic system in here. So by this what I would call it, so this bis-benzyl bridging unit, and by this you can really achieve the regioselective synthesis of these adducts quite nicely. And then you have two possibilities. You can either cleave the ester or you can cleave the tertiary butyl ester under acidic conditions, or you can just hydrogenate over here just to have the two possibilities. And for the example I'm now showing to you is just we cleaved here over the ester under acidic conditions first, and then decarboxylated here. So then we have only this compound being left over, and then you do just a hydrogenation, or by some other means you can just cleave off this residue, and then you have very selectively synthesized only one of these regioisomers of the bis marked fullerenes. Again, we did Ugi reactions, we did Passerini reactions, so we knew the conditions. Again, aniline has to be used, and really quite some reasonable amounts we could use these bisfunctionalized modified fullerenes very easily. I mentioned to you that we use this or that we wanted to study them as reagents in the photodynamic therapy. And what you check in this case, so what is done as a regular or as a monitoring reaction. So you have this benzofurane, so this modified benzofurane, and you treat it with singlet oxygen, and then you have this cycloaddition being, or this cyclized, or this cycloaddition product being formed, and you can watch it nicely by watching at the UV spectrum. You can just interfere with this reaction when you just have photosensitizers being added, and we, for instance, have added these 5 modified fullerenes, and these studies have been done at the UFSM, very close by your place. And what has been found out that this compound, so where you have this dipeptide attached to the fullerene, is actually best. So you have a very, 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 very nice quantum yield. So if you just have as a standard the fullerene with a quantum yield of 1, then this comes very, very, very close. Also, the for instance, compound 9, which has this really nicely solubilizing side chain, also gives you some reasonable quantum yields. So we checked all of these things by the decrease of the UV signal, and by this you can then just calculate on the quantum yield, and this gives you a measure how successful these compounds can be used in the future. So further studies need to be done. So we haven't done any further studies really on cells or on living organisms by now. Maybe we are continuing with this in the past, but I just wanted to show that these modified fullerenes can be used also for human health issues. Just to play around synthetically with these molecules even more, so you can also do some let's say post UGI modifications, and what you see, for instance, if you use this modified anilines, so carrying a vinyl group in here at the para position, then you can do some palladium catalyzed coupling, so you can add this carbohydrate unit, which also gives you or gains a lot in solubility. You can also have this alkyne modified aniline, and you can imagine what kind of chemistry we have been done. This is just the classical click reaction. So by this, you can just attach all sorts of side chains in a post-UGI manner. So you can really improve the wealth of these molecules by setting up these further reactions. And chemistry gives you so many opportunities and so many chances. So we have a lot to do in the future. So the conclusion is actually that for the first time we carried out UGI and Passerini reactions, so isonitrile modified multi-component reactions on these fullerenes. And they showed really some promising results as photosensitizers. You can really improve the applicability, or let's say the chemical space in which these compounds can be used by having these multi-component reaction modifications. Yeah, so a lot of things can be done. Yeah, this has been published some time ago. And with this, I would like to thank the people who have been involved in this in these very, very nice studies, and also would like to thank the foundations who gave the money. So it's a CNPq, and some years ago you had this very nice program in Brazil, Science Without Borders. So by this, Brazilian PhD students were allowed to come to, or were allowed to going abroad, and some of them ended up doing their PhD studies in Halle in Germany, and for this we are very, very grateful as well. So I think I've shown you that carbon nanomaterials can be used for agricultural reasons, so for plant health. They can also be used for human health studies, and I think a bright study is ahead of us to use these materials very nicely in the future. But as well, we have to be sure about the positives, but we also have to be sure about the negatives. Yeah, so there have been some reports that some negative side effects have also been could also be obtained and determined by using these compounds. So we really have to find the right way to have everything more directing to the positives than to the negatives. But it is possible, and I'm very, very optimistic when it comes to this. Yeah, I'm now finalizing with the last picture of Halle. So this is the sky disk of Nebra, which has been found very close to Halle. Maybe you've seen it on some jewelries or so. This is the eldest depiction of the cosmos, and whenever you come to Halle, please go to the museum in which this one is displayed. It's a very, very nice display of former science. Right now we are in modern times, modern science, and I'm very happy to be allowed to give this lecture, and I would like to thank very much for your attention. Vielen Dank. Obrigado.
Thank you very much, Professor, for your excellent lecture. It was definitely a valuable presentation for everyone here. Cristiano. So, anyone have questions?
I have one, if I can. Can you hear me, Benny?
Yeah.
Yes.
I'm curious regarding your UGI reaction with the fullerenes, do you have some? You could observe some stereochemical control in this peptide generated? Consider if you have a substitute other than formaldehyde, there is some stereochemical induction, or it's
No.
just one-to-one as always?
No, as always. So no, there's no stereochemical induction. So we did the reactions with a couple of aldehydes. Formaldehyde or paraformaldehyde works nicely. Acetone also worked in certain cases. So in these cases, you do not run into any stereochemical problems, as you know. Benzaldehyde works also nice, but no, we didn't obtain any induction. Yeah, so we have to see if the stereochemistry is a limiting case, for instance, in human health applications. But this we haven't done yet. Yeah, so the typical problems you obtain when doing these UGI reactions.
There is a question here. Someone asked you in the chat, Benny, what do you think will be the most the biggest challenge in this research from now on?
The availability of the carbon nanomaterials. Yeah. So if you provide these in agricultural business, you know that the availability and the price is really a big factor, and you need to have these carbon nanotubes in bigger amounts and cheaper than it is actually at this moment.
Okay, thank you. There is, I think, there is no other. Tell me, how is Gabriela?
Doing fine.
How's Gabriela doing?
Yeah. No, it's really doing fine. I think everything is moving forward, and I also talked to Darius on the use of these of the light scattering. So I think we can so maybe in 1 or 2 weeks we can also start these investigations.
Yeah. That's nice. Okay. Well, then, if there is no questions anymore, Benny, thank you again. Thank you again for your time. Thank you for your talk. It's very nice. It's really nice to see you and hear you all. And thank you everyone for being here. Obrigado a todos por comparecerem e compartilharem do nosso Nano Network. E nos vemos em breve. Thank you, Benny, again.
Okay.
See you soon.
Okay, see you soon. Obrigado. Thank you very much.
Bye.
Bye.